create a disease-resistant animal by modifying its genome? Well, in fact, with the advancements in the world of genetic engineering, it may be possible. Scientists have discovered this gene targeting system called the transcription activator-like effector nickase, which allows them to build a protein in the DNA that can target and modify a specific sequence, or more precisely, can create a single strand break in the genome quite nicely. Heibo Wu and his team of colleagues employed this talent system, which enabled them to modify the genome of cattle for increased resistance to the infectious disease tuberculosis. First, the team tested whether there were nuclease-induced insertions and deletions at the targeted site of the genome by PCR amplifying the targeted site and transforming them into E. coli colonies. From this, they found that from the 6.13% of the E. coli colonies, there were deletions and insertions present. This indicated that they had created their very own artificial gene targeting system. They utilised this system to insert a gene derived from a mouse called SP110, which they found mediates innate resistance to tuberculosis. Wu and colleagues then depicted their targeting strategy using a bovine endogenous macrophage receptor promoter. They found this directed the expression of the SP110 gene. After the successful insertion of the SP110 gene, Wu and colleagues then selected 9 of the transgenic cell colonies and used them as donor cells in order to clone the transgenic cattle. The team did this by reconstructing embryos and culturing them in vitro, which developed into blastocysts and were transferred into the oviducts of recipient heifers. They recorded a total of 23 calves that were born, in which 13 survived into adulthood. They then integrated the SP110 gene at the targeted site of each of the calves, and found the transgenic cattle were heterozygous for the SP110 knock-in. In order to determine whether the SP110 transgene could be passed on to offspring, the team acquired 3 transgenic offspring calves from the transgenic cattle by means of AI. From their southern blot analysis, they found that one of the calves were heterozygous for the SP110 knock-in. This indicated that the SP110 site-specific knock-in for tuberculosis resistance in cattle is heritable, which suggests that this gene can be passed into one generation without actually having to genetically modify the cattle. This approach of modifying the genome of cattle used by Wu and colleagues is absolutely applicable to the contribution, control and prevention of bovine tuberculosis, and is also useful in the insights of breeding animals for disease resistance. In the last couple of years, several studies have utilised the talent-mediated approach to successfully knock out genes in cattle and even pigs. However, no scientist to this day has actually successfully engineered a specific site of the animal's genome to increase disease resistance. Until now.